Okay, welcome to this final session of our 8th MCHRI Symposium. I'm Kara Meister. I'm an assistant professor of otolaryngology in the Division of Pediatrics, and I am honored to introduce this cohort of speakers. We do have one slight change in the agenda. Speakers 2 and 4 will flip-flop, most likely. Stay tuned for about 7 minutes while we decide. But first, we have Dr. Tippi Mackenzie, who will be our first speaker in this session. Dr. Mackenzie is a professor of surgery at University of California, San Francisco, where she's the Eli and Edythe Broad Center of Regeneration Medicine and Stem Cell Research at UCSF. She's the co-director of the Center for Maternal-Fetal Precision Medicine and the John Bowes Distinguished Professor of Stem Cell and Tissue Biology. So we're so honored to hear her incredibly groundbreaking work. Thank you.
Thank you so much for the kind introduction and the invitation to speak here. I like to tell people I met my husband in the Stanford Med School courtyard, so it's a really special place for me. Love being back. Okay, these are my disclosures. We get research funding from some companies whose products might be worthy of testing in mice or sheep in utero. So I first got introduced to the concept of fetal surgery when I was a medical student. In 1996, I did a visiting rotation at UCSF with Dr. Mike Harrison, who had recently invented the field. And over the past couple of decades, it's expanded to go from open fetal surgery to laparoscopic, or what we call fetoscopic surgery, to catheter-based interventions. It's performed all over the world, including obviously at Stanford. What's been around even longer is the concept of fetal blood transfusions, where you can do an ultrasound-guided injection into the umbilical vein. In experienced hands, it takes about half an hour, and it was used since the 1960s to deliver blood in cases of RH discrepancy between the mother and the fetus. And of course, if you can put basically an IV into the fetus, then you can deliver any other kind of medications, including stem cell transplants, protein replacements such as for lysosomal storage diseases, or even gene therapy or genome editing. So I'm calling these collectively fetal molecular therapies. And of course, you can't treat what you haven't yet diagnosed, and so the fields of diagnosis and treatment very much walk hand in hand. Fetal surgery came about because of ultrasound diagnosis of anatomic malformations, so that's why pediatric surgeons like Carl Sylvester and I have been involved in the field. Sometimes we can diagnose genetic diseases based on ultrasound manifestations, and the most common of these, there's a constellation of symptoms called hydrops, where the fetus has ascites and pleural effusions, and most often these are due to genetic diseases like alpha thalassemia and lysosomal storage diseases. So it's no surprise that we've chosen, like, those are the two first genetic diseases for our molecular therapies, because they literally walk through the door with hydrops, as opposed to the base of the iceberg, which is patients with severe early onset genetic diseases who of course don't manifest on ultrasound, which is the only screening we do in pregnancy. You know, that's I really enjoyed the talk on the newborn screening, but we don't do that routinely in utero, so we're awaiting ultrasound manifestations. And it's very much a yin and yang, so although there is prenatal carrier screening, even whole exome sequencing available, patients don't opt for the screening if there isn't a treatment that could be given if they've already decided to keep their pregnancy. So it's very much a yin-yang. So candidate conditions, of course, we're not making designer babies. This is for severe early onset genetic diseases. I've listed some of my favorite ones there by kind of disease category. I'm sure there are others I'm missing. I need to put epidermolysis bullosa on there. I keep forgetting, but you know, this is, the list goes on, right? And why not wait until birth? So of course, we want to prevent irreversible organ damage. That's how the entire field started. but it also turns out that biologically the fetal environment offers some unique advantages such that the therapies would probably work better. So early in gestation, the blood-brain barrier has not completely formed. This is a very complex interplay. It's not just that it's open or closed, but obviously there are cell types and regions that are important there. We have a collaboration with my colleague Betsy Crouch in our Division of Neonatology who's studying this in detail. The fetus has a really unique immune system. It used to be thought that, oh, the fetus doesn't have an immune system, but of course it does, and it's quite sophisticated, and it's wired for tolerance to self-antigens, and it turns out also non-inherited maternal antigens as a result of maternal cell trafficking. So that could be a benefit of tolerizing to new proteins. Stem cell populations, for example, hematopoietic stem cells are more accessible where they are developing in the fetal liver before they go hiding out in the bone marrow. And the fetus is tiny, so you're talking about a cost of goods or a cost of manufacturing for a 500-gram fetus. So it's a fraction of what it would cost an adult or a newborn. So here's the vision, so that we would do early genetic diagnosis. We would confirm that it's a severe disease, so that by definition means we have to know the genotype-phenotype correlation, because we're diagnosing a disease based only on the genotype, hopefully before there are any symptoms of disease. Non-directive counseling is the bedrock. You have to be able to discuss the various options of the pregnancy. Then the parents choose the best treatment that can be given before or after birth. And here's the treatment menu, right? Depending on the disease and depending on what's available, anything from stem cells to protein replacement, antisense oligos, gene therapy or genome editing. And there's also the important and emerging field of medical treatments given to mom that I don't have time to talk about, but it's something I'm watching very carefully. And these treatments can be given under local anesthesia. This is a picture of one of our stem cell transplants. So just local anesthesia and some sedation to the mom if desired. Ultrasound guided, requires steady hands, but quite safe and experienced hands. There should be less than a 1 to 2% risk of adverse outcomes. And then obviously long-term monitoring of the fetus, the mother, and then the neonate. So the field has been around for a long time. The very first stem cell transplantation was performed in 1989. Dr. Maria Grazia Roncarola was involved in that first paper for bare lymphocyte syndrome. And more recently, we've done a clinical trial for alpha-thal major that I'll briefly touch on. Protein replacement is a relative newcomer, started with Holm Schneider's work in X-linked HED. And now I'll tell you guys a little bit about our enzyme replacement therapy clinical trial. And I think the real future is gene therapy or genome editing, for which there's been small and large animal work for over 30 years, but no clinical attempts yet. So let's go with alpha-thal major. So we were all taught in medical school or everywhere that alpha-thal major is a fatal disease. So this is just a sort of backup. So as adults, we all have alpha and beta hemoglobin tetramers. As fetuses, we make alpha and gamma hemoglobin. But if you're a fetus with deletions of all 4 of your alpha globin genes, then you're making these hemoglobin BARTs that are gamma tetramers. And so in the textbooks and the review articles, this form where you're missing all 4 genes is called hemoglobin BARTs, hydrops fetalis, and it's lethal. Fetuses develop hydrops, which is just a harbinger of their anemia. So the very first patient that we saw with alpha-thal major is Eliana, and Eliana's parents were told that their fetus that presented with hydrops was destined to die, and they did an internet search. They found our clinical trial that had just started, where we proposed to put to transplant maternal cells, and they said, I think we're going to go to San Francisco. What we had discovered when people had reported before is that patients do well with serial in utero transfusions. So they can survive, but there are very few survivors reported. And postnatally, the disease is similar to beta thalassemia and can be cured with HSC transplantation. So recognizing all of this and then recognizing the biology that the fetuses, their immune system learns to tolerate non-inherited maternal antigens. We decided to transplant maternal cells in 10 fetuses with alpha thalassemia. So we obtained an IND to do this, and we transplanted 6 patients over the course of about 5 years. I won't go into all of them in detail, but what I can say is it's not a fatal disease, which is the most important thing we learned. They all had blood transfusions in addition to the stem cell transplant. These are the 6 that we transplanted, and I'm really grateful to all the families for allowing us to share names and pictures, obviously with consent. They were all born at term, but the engraftment was very, very low. And here I'll back up by saying we don't know how to condition the fetus. So when you do a bone marrow transplant after birth, you're doing conditioning, immunosuppression, but we can't do that. So what we're hoping is that the fetus is tolerant to maternal cells, and then you just give a 100-fold higher dose of cells. But we discovered is that alpha-thal, although theoretically this process could work and it's worked in dogs, alpha-thal is probably the worst possible disease to test it on because there's an expansion of hematopoietic stem and progenitor cells in alpha-thal. So in a sense, we chose the wrong disease. And what I'm showing you here is a flow cytometry plots of some of our alpha-thal patients as well as a control patient. And you can see that there's a huge expansion of CD34 cells in the alpha-thal cells. So we chose alpha-thal because the FDA wanted us to choose a fatal disease, and so we showed that it was safe and feasible to diagnose patients in utero, but we chose absolutely the worst disease. And so what would be the best disease is Fanconi anemia. And so now we're really excited to collaborate with Agnieszka Czechowicz here because, of course, there's a competitive advantage of wild-type cells in Fanconi anemia. So even if you get like a 0.5% engraftment over time, that could go up. And this is a paper that's from Agnieszka's lab that she published last year in a mouse model. The red bars are Fanconi anemia mice that got an in utero stem cell transplant, and you can see compared to wild-type mice, the levels of chimerism continue to go up. So that's the next IND. We've been collaborating with Agnieszka to submit an IND for a two-center trial, both at UCSF and at Stanford, for in utero stem cell transplantation in FA. So switching gears. So the next thing we're going to do is to develop a gene therapy for the patients who are born with alpha thalassemia. So this we're calling the Great California Bake-Off. So either lentiviral gene therapy, this is with Don Cohn, or inserting hemoglobin A into a genomic safe harbor. This is with Kyle Cromer, who's trained with Matt Porteous, who's now at UCSF and is doing incredibly well. Or upregulating the embryonic version of alpha globin, which is zeta globin, which is a project that we're doing in my lab. Don and Ava have won the bake-off, and they're preparing the IND for their ex vivo lentiviral gene therapy trial. We're hoping that the insertion or the upregulation could be direct in vivo approaches, and we remain excited about working on them. So moving on to enzyme replacement therapy, this is another clinical trial where basically we're giving in utero enzyme replacement therapy to lysosomal storage diseases, all of which, so the 8 are listed here, they all have FDA-approved postnatal enzyme replacement therapies. And the way that the patients get diagnosed, usually in families where they know that the parents are carriers, they've often had another affected child. We give the enzyme. You have to give it every 2 to 4 weeks because of the half-life of the enzyme, right? It's not a curative treatment. And then we do postnatal standard of care, and we're doing yearly follow-up. So this is really a phase 1 safety and feasibility study, and we're trying to see whether there's a benefit of treating in utero, and this is sort of a way to sort of dip our toe into the field before we can do something more definitive like gene therapy or genome editing. But we're learning a lot from it. For each patient, we have an enrollment advisory board to make sure we're only enrolling the severe genotypes of these diseases. So the first time we did this, it was in 2020, in the height of the pandemic. The first patient was Edythe. She's actually Canadian, and they couldn't fly during the pandemic, so we collaborated with our Canadian colleagues. She got 6 doses in utero, and it worked remarkably well. We got very lucky here in that Pompe has a prenatal ultrasound cardiac manifestation. So here's Edythe's previous sibling, prenatally diagnosed, and on the 34-week echo, you can see the septal thickening here, whereas Edythe, after a couple of doses of in utero ERT, had a completely normal heart. The CK levels have also been normal. The gray lines here are 4 patients with infantile onset Pompe who were diagnosed on newborn screen, and you can see even at birth, before they've developed any symptoms, the CK levels are above normal. We've since treated 3 patients with infantile onset Pompe, and so these results have repeated. So we've seen rescue of the predicted cardiac hypertrophy. In all cases, there was a previously affected sibling. Pompe is a muscle disease, so there was often hypotonia, but there was no hypotonia seen in our patients. And this is kind of a nerdy detail, but I'll just put in that 2 out of the 3 are the CRIM positive version. All of these patients develop immune reactions to the enzyme replacement therapy, and they have to be given methotrexate and rituximab, what's called immune tolerance induction. We were able to avoid that in 2 out of the 3 of these patients. I'll just quickly go over a couple of these other patients. So patient 2 had MPS II, which is an X-linked disease. One of the things we do is when you put the needle in, you have to get a little bit of blood back. And so in that blood, we check enzyme trough levels. And so that's our way of doing a little bit of PK. And so the enzyme trough levels of the fetus here are quite low, which is good. That tells us the fetus is truly affected. And then we also look for dried blood spot biomarkers, and you can see here in the fetal blood, these biomarkers are going down over gestation compared to the maternal ones that are, start out low. And then this is the newborn screen dried blood spot, and you can see that the treated patient tracks with the normals as opposed to all of these patients who were diagnosed based on that newborn blood spot. So there's a bit of a hint of efficacy. I mentioned the blood-brain barrier. It's not as simple as I mentioned that it's like open or closed, but there was a patient with neuronopathic Gaucher disease we recently treated who presented at 20 weeks gestation with clenched fist, which can be a sign of a corticospinal tract injury. And we saw after 3 doses of the enzyme replacement therapy that the clenching resolved, and the fetus gave us a thumbs up sign before the 4th ERT. So postnatally, the MRI was normal, the fetal MRI was normal, and initially the thumb moved, but it was somewhat adducted. I will say this patient is now 6 months old, and of course is now starting to show signs of the neuronopathic Gaucher because the postnatal enzyme is not seeing the brain. So this is, while it's a proof of concept that you can treat the CNS disease in utero, it's really motivated us to do an actual curative treatment in utero. So we've treated 6 patients so far, and they've all had between 3 to 6 infusions, and there have been no adverse events related to the medications, and we see hints of efficacy. Obviously, a small data set right now, but it's not great, of course, that every patient needs in utero procedures, and as I mentioned, we're really motivated to find a definitive treatment. Which takes us to gene therapy. I don't have a lot of time left, so I'll just say we're not treating embryos. This is not embryo editing. We're trying to do somatic cell gene therapy or genome editing. I think this is a really important distinction. And theoretically, any modality that's used postnatally that could be used in utero, and we're really trying to look at things that have been given to infants postnatally for use in utero. So while we are doing experiments with antisense oligos and genome editing, I'll just say, the next thing we're working on for clinical application is AAV gene therapy, because that is so commonly used now, in addition to, Zolgensma for SMA, in addition to other things. There's a very long history of prenatal AAV gene therapy in animal models, but no clinical translation yet. This is a sheep study we published recently demonstrating expression of GFP after an AAV9 GFP vector. So we recently submitted an IND to do this using an AAV9 viral vector that's been used by Cindy Tift and her colleagues to treat GM1 gangliosidosis, which is a very severe early onset lysosomal storage disease. It's an academically owned vector. It was safe, and they had demonstrated efficacy in animal models, but unfortunately, in these children, some of whom were dosed early in life, they really did not see efficacy, which is sort of the flat gross motor skills that you're seeing. And so we thought this was and there is ample evidence of prenatal onset of disease. And so we thought this was a compelling situation in which you have a very early onset disease. There's already a vector. It's tested in infants. It's safe but ineffective. And so there's a lot of rationale for doing this. So fingers crossed, obviously, for IND application. But of course, there are other AAV9 vectors that are currently used in infants, for example, with SMA, and ones being made for GM1 and GM2 gangliosidosis. And then, I think we're all very interested in base editing and genome editing. So I don't think I have time to show you these, but just to say we're doing a lot of super fun experiments in mice, testing lipid nanoparticles, carrying genome editors. And this, everything green that you see there is a genome edited cell in a CRI mouse model. We're also working with Cas9 mouse models. And then the CNS diseases, I think, are some of the most compelling for in utero therapy. And so we're collaborating with Richie Chen, who's got a CNS tropic lipid nanoparticle. And this is kind of the last thing I'll point out is this is a CRI model. So these are edited cells in the subventricular zone. And when you wait a couple of weeks, they migrate through the CNS, and you see them throughout the brain. Whereas, if you do this postnatally, you only see the needle tract. And of course, we do fetal neurosurgery, right? We do the spina bifida repairs. People are doing ventricular shunts in utero. Look at that. This was done in utero. And so we're practicing this in the fetal lamb model with our fetal neurosurgeon. We work very closely with ethicists to make sure that we're doing this in the correct way and surveying patients to make sure that they would want this therapy. So, we're working on a lot of different modalities, from stem cell transplantation to genome editing, trying to de-risk therapies that are already tested in children, but also in the future trying to find new therapies designed for fetuses. So it's an army of people, so grateful to work with, and especially our patients for their participation in research. Thank you.
Tippi, that was awesome. I mean, absolutely just amazing.
Thank you.
What struck me was just the last set of slides with the brain.
Mm-hmm.
So it seems like because it's so much neural development, you can hit the progenitors early. Can you talk a little more about what would be the predicted, in translating to humans, when you might do that? We heard a lot this morning from David Roach and others about you can diagnose conditions early, how that might work. Just the timing.
Yeah, I mean, I think the timing, honestly, and David, I'll have to just talk to you and get your input on this. I don't know that we diagnose these patients early enough to really hit the stem cells at that stage in development.
Yeah.
So I think I don't know how well it's going to translate to humans, but as I mentioned, the diagnostic and the therapeutic arms go hand in hand.
Right.
So, if you have the treatment, then perhaps patients are going to start getting tested earlier and earlier, and you might be able to intervene early.
Right.
And then hand in hand, maybe you don't need to hit the ventricle. Maybe you can develop something that crosses intravascularly or give through the amniotic fluid.
Right.
So, I mean, I think,
No, but
there's a lot of
very
development
impressive.
to be done.
In the GFP model, it looks amazing.
Thank you. Thank you. Hi.
Tippi, that was a great overview of the wonderful work you're doing. I wonder, with the recent knowledge about links between maternal conditions or maternal ways to treat fetal conditions. And you mentioned very briefly medical therapies you're not doing, but also recent knowledge about placental defects that are associated with fetal defects. And when you fix the placenta, you fix the heart. And it's been shown in mice in several models. And now there's some discussion about placenta-brain connections and maybe fixing the placenta as a way to fix brain conditions. Is this something you're interested in, potentially translationally to take to patients?
Oh, very much so. I think placental gene therapy is a really hot topic, and it might end up honestly being one of the first gene therapies involved, because it is, you only need it for those 9 months, and then, if there's an adverse event, then it's gone. So I don't personally work on it, but a lot of my colleagues are working on placental gene therapy, for example, for IUGR. I think there's enormous potential in that field, absolutely. Thank you for the question. Thank you. Okay, thank you.
Our next speaker is one of our own, Dr. Matteo Molle, who is also on our education committee. He is an assistant professor in the Department of Obstetrics and Gynecology and runs a lab as well. So we're excited to hear his talk about dissecting interactions between the embryo and the endometrium during human implantation. So a perfect setup on the placental question.
Thank you so much. Hello, everyone. I'm very excited to be here, and thanks for the introduction. So, my name is Matteo Molle. I joined quite recently, about 2 years ago, Stanford, in the OBGYN, and actually quite recently became part of the MCHRI education committee. Very excited about this, and so I thought about taking the opportunity to tell a little bit about what we do in the lab and why we're excited about working at this very niche stage of development, which is human implantation, and trying to really understand the first communication between the mother and the fetus, so embryo, at that stage during implantation. Okay, so the journey starts actually in Cambridge. So going back to David as well, where we share a beautiful view of the college here. I'll try. I will fail. Okay, here there is a laser. And actually, a little bit, my journey, especially scientifically in terms of research, starts really with this milestone event, the birth of Louise Brown in '78 with IVF, pioneered here the picture of the first IVF conceived baby by Bob Edwards and Patrick Steptoe. And here is Bourne at the first IVF clinic not far away from Cambridge. And it's really this event, and a little bit, maybe I was based in Cambridge, and a little bit I wanted to continue part of the journey in the fertility treatments that brought me to the stage of studying implantation. So, looking a little bit about the statistics, it's estimated by the WHO that 1 in 6 people in the world are affected by infertility, which is really astonishing. And as we know, IVF is one of the key tools that has been indeed pioneered in '78 by Bob Edwards and Steptoe to allow people and couples struggling with infertility to build a family and have a child. And up to now, this is I tried to pick up the most recent statistics I could find, and about 10 million babies have been born by IVF in the whole United States, which is really impressive, considering also the short time of the development of this technique. However, as also Ruth and many others introduced today, we know that there is also limitations in the success rate of IVF. I'm sure we all know how many times cycles of IVF fail, and Ruth as well talked a lot about the chromosomal abnormalities and many other factors that can influence this. And we know also how costly and expensive it is for family, but also emotionally failure in this procedure can bring. And this is from the Financial Times, just recent fertility rates projections in most of the developed countries, and also Carlo also discussed today, they're all going below the replacement level. And also obviously we live in a completely different society, which contributes as well to this decline, because planning a family is now shifted to older age. And I want to start here. So this is actually one of the article from Bob Edwards, where he says, what is the latest barrier in the assisted reproductive technology? And it's implantation. This is what it defines. And I want to go a little bit about these statistics, which is not very easy to, has been lots of work from different people to quantify how many embryos are lost between, basically, if you look at this is a pyramid of conceptions. It's estimated that about 30 to 60% of embryos are lost specifically at the interface between the pre-implantation, before the implant into the uterus, to the early post-implantation, giving rise to this, what is called preclinical miscarriage, so before even any clinical detection. And then it's estimated about 10% of miscarriages occur later, classified as clinical loss, and about 30% of those embryos will result in live birth. So that's really, as we can see, a big bottleneck for human reproductions. And actually now they're trying to quantify this also for natural conceptions. And there are suggestions that there is quite similar as well. So really implantation is really a barrier for human reproduction. And so that's the process of implantation, IVF in a schematics. So what they do in an embryology lab. So they undergo the process of fertilization in vitro, grow the embryo, and then eventually you need to transfer the embryo inside the uterus. And this is where we lose control. We don't know what happens, and there where we face most of the embryo loss, because at this stage this can give rise to a successful pregnancy if the embryo successfully implants, form the placenta, and carries on, or a preclinical miscarriage. And this is really what we are interested in this stage, really, when the embryo, at this stage is called the blastocyst, reach the uterine tissue here, so the uterine cavity, and the uterine tissue called the endometrium starts these interactions, which is really the first embryo-maternal interactions to occur, the very first contacts, and the embryo needs to invade very deep, actually, because our implantation is why specific human implantation is very interstitial, and then enter into the tissue where it will build up the placenta at the interface with the maternal vessels. So this is really what we wanted to understand, that this is a little bit of our general goal is, if we try to understand the basic mechanism of communication between the embryo and the endometrium, hopefully we can translate this basic understanding into clinical interventions to make the process more efficient and so overcome the process of implantation barrier, this implantation failure. So one of the major difficulties that we encounter, and this is where the journey started, is how do we study this? Because animal models, we know they are very different. Even primates per se have are different from human placentation. Just older primates like bonobos and gorillas have similar placentation to us. So the first challenge is we need something that can recapitulate the maternal uterus and then simulate the process of implantation in vitro. So the first challenge that we face is to recreate something in vitro that could recapitulate some of the complexity of the human uterus. And to be able to do this, we started with the recruitment of many patients' biopsies of the uterine tissue. So these are endometrial scratching. And what we did is we isolated the main cell types, in this case the stromal and epithelial cells, propagated in 2D, considering that the initial biopsy is quite small. So we propagate, we grow them, bank them. And then we use reverse tissue engineering. So that's a lot of work summarized here about basically encapsulating the cells back into a 3D tissue engineer system based on hydrogel, where we work a lot with the chemistry, the degradability of this, and specifically also the composition, because it contains specific extracellular matrix molecules, to try to recapitulate a little mini tissue of the human uterus in this transwell dish. So here is our model. So if you go from the top to the bottom, you can see there is an epithelium layer, and then going deeper and deeper, you can see the stromal. The stromal cells are the fibroblasts of the deeper tissue. And this is a little bit a comparison. This is the in vivo tissue on the top from the actual biopsy, and this is what we engineer in the lab. And you can see the similarities of having indeed an epithelium on top and a stromal compartment. And now we're working more and more also to achieve much more depth in terms of the tissue, which has been one of the major obstacles so far. So of course we were happy to see that starting from the patient, we can isolate the cells and then rebuild in vitro the uterine tissue, the patient specific, but that's just anatomically resembling the human uterus. So we wanted to have evidence whether this tissue could be hormonally stimulated and responsive to the hormonal stimulation, similar to what happened in the menstrual cycle. And this is what we did. So there are many protocols. So we just followed basically an initial stage where we give estrogen to simulate the initial proliferative phase of the menstrual cycle, followed by progesterone and estrogen, because we wanted to reach this stage when there is the second peak of estrogen and progesterone, so the mid-secretory phase when the embryo reaches the uterus and potentially undergoes implantation. So we gave this hormonal stimulation, and we could see that the resemblance, so the massive changes occurs in the tissue. So that cells were responsive despite they've been isolated and taken out of the primary tissue. As you can see, the stromal cells change dramatically their morphology from elongated, they become cuboidal, epithelioid, but most importantly, they acquire secretory phenotype, and one of the secretion is IgBP1, which is a key marker. As a second part, and unfortunately I had to condense a lot in few slides, is on the top of the epithelium we also observed the formation of invagination that at that time we speculated could be similar to the glandular invagination of the endometrium. And actually after hormonal stimulation we could see that the cells in close proximity of this invagination starts also acquire secretory phenotype, secreting this key molecule pepglycodelin, which has been described to be one of the key components of what Graham Barton in Cambridge defined as the uterine milk, which I really like as a nomenclature, because it's the collection basically of secretion coming from the endometrial glands that nourish the embryo before the placenta becomes functional. And we could see as well, so you can see here, this is a time lapse over 12 days, where you can see these holes almost kind of, that becomes this invagination and then become very convoluted over progesterone and stimulation. So really resembling the deeper glands that we see in the primary tissue. And we also see a massive shift on top. So by just looking at the epithelium on top, we see a massive shift as well in the ciliation. So you can see here the formation of very distinct cilia on top of the epithelium. So here you can see cross sections, but also enrichment of these cells in pink with FOXA2, which really are cells which acquire more secretory capacity and versus basically the luminal versus the secretory part of the endometrium. And we did lots of quantification about this, and now our manuscript is finally accepted. And this is where we're going forward. So this is still ongoing work, and it's really thanks to the amazing collaboration with Lucy, Ruth, Anthony, and Alex and Berta. Is, of course, we're still missing many key cell types of the human endometrium that we want now to isolate and engineer. One is the endothelial cells, and the scope is really to isolate still from the same patient biopsy the endothelial cells to build the maternal vessels. I thought it was going to be an easy experiment, but they proved me wrong. So we're 12 months and we're still struggling a lot. It's not an easy cell type. And I bet we even have started with the immune cells, and it's going to be even worse that. So I'm not touching the immune cells until we solve the endothelial cells. But the idea is to really build a basic endometrial tissue that resembles the patient, because it comes from the patient cells, and then this can be used as a platform for diagnostic therapeutics and in general for us to understand the process of implantation. Because really what we're really interested indeed is what happened at the interactions between the embryo and the endometrial tissue. Now that we have this platform in 3D, complex. To a certain extent, obviously, it's not the primary tissue, but that's the best that we can do up to now. Is that what about now? Can we study the process of implantation? Can we achieve actual implantation in vitro? I mean, in 78 they achieved in vitro fertilization, so I'm hoping now we can move to in vitro implantation, which is really the next goal where we're trying to go. And so what did we do is, so thanks exactly with the collaborations with Liesel and Ruth, we have access to an amazing resource here in Stanford, which is the Renew Biobank, which is a collection of several embryos donated by IVF patients. And what we do exactly what they would do in the clinic, in the laboratory. We will thaw the embryo, and then instead of transferring into the uterus, we transfer into our system, and then we watch over time the process. And you can see here the human blastocyst. This is day 5. And this is the zona, the protective shell, and you can see it hatches after 24 hours, so it literally pops out of this protective shell. It massively grows over time, and you can see that also this is the adhesion of position, so it's really oriented towards the side of the maternal endometrium to eventually attach by this deflation. I know here it doesn't look good, but I'll show you after a few slides that it is actually beautiful, the morphogenesis that occurs of that horrible structure that you see there. And we're trying to quantify many things that we don't know about human implantation. For example, we're trying to quantify the stages when each of these stages occurs, implantation, the first adhesion. And here we have about 117 total of embryos. So we're trying to really build up a basic understanding what time human implantation occurs, if there is any correlation. We have also embryos with chromosomal abnormalities and embryos, healthy embryos, untested embryos, and we can see as well the loss when the major loss occurs, so when the major degeneration of these embryos occurs now that we can follow over time. And here also you can see we do screening as well of key secreted factors such as human chorionic gonadotropin, which is the primary factors of pregnancy. So you can see that basically these petri dish are basically pregnant as soon as the embryo starts to contact the maternal tissue. We also do screening of other pregnancy-related tests. And really here what we wanted to understand is, because this has been up to now inaccessible because it happens inside the uterus, we haven't really advanced anything in this field. So now potentially bringing this into the lab, we have accessibility to understand the basic, but also to develop intervention in this field. So this is how it looks. As I promised, it looks quite nice. So you can see this is the embryo proper. All of this surrounding in pink is the placenta. So you can see the extent of the human placenta day 14. So this has been done in the UK, because in the UK we can culture the embryos until day 14. In California we can culture the embryos until day 12 because of stricter legislation. And surrounding you can see the maternal tissue. So these are all the endometrial cells of the mother. And you can see even the projection of these placental derivatives. And we have gone through a lot of analysis of how the initial stages, the initial building blocks of the human placenta are set up, how almost anchors into the maternal tissue invades and go deep. And we're trying to address now really understanding the interactions. So this fetal-maternal interface, these initial interactions between the embryo and endometrial tissue from many different points. So one, of course, is from a transcriptomics level analysis. So you can see here, so here we have a total of 17 implantation sites. It may not sound a big number, but it's actually very, very difficult to get this in the human embryo, field. So all of this is the endometrial tissue, all of this is the embryonic tissue. So we can split and we can understand basically how these two tissues interact. And there is a lot of bioinformatic analysis as well that we're implementing to try to predict and understand what receptors are expressed on one side and what are the ligands expressed on the other side, and how this potentially could interact. And so these are all prediction based on their expression level. And really what we wanted to understand is how the two tissues and the different subtypes of the placenta and the different subtypes of the endometrium interact and communicate with each other. And this graph I don't like, but that's the best we could come up because it's very dry. But I mean, the content is very beautiful because what we did is we identified some interactions that are specific at the implantation stage, so at day 14, so first 2 weeks basically of human pregnancy, and then projected against the first trimester abortion, so that we could basically pinpoint what are specific interactions that occurs only at day 14 versus the first trimester abortion. And as you can see, there are many shared interactions, so that means that something that is established early as implantation is kept throughout the first trimester. But you can see as well that there are some specific implantation specific and some that only are first trimester. So really trying to dissect temporally as well how the first interactions at implantation towards the first trimester, they diverge or converge with the different cell subtypes that we have. And we're addressing this as well, obviously, from, as I mentioned, using very different techniques. One is live imaging. As we know, implantation is not a static process, it's a very dynamic process. And for this, Nicole here in the audience has managed to go through a completely crazy, insane experiment that she took on board, luckily, to try to keep the embryos alive under the microscope, which is not an easy, and stain them as well with fluorescent dyes that are not toxic. And so there is a lot of optimization behind all of this. But the nice thing is that we could see how the initial interaction occurs and how this implantation grows over time, and we can do now lots of quantification. So this is one of the ongoing project of Nicole, PhD, really trying to understand the dynamics of this process. And another project, just to give an overview where we're going as well to address this. Obviously, these are cell-cell contacts, but there are also paracrine signals between the embryo and the maternal tissue. So Zach, another PhD student, is actually working on trying to understand what are the factors secreted by the mother, what are the factors secreted by the embryo, and how these communicate. So we're putting together a completely big unbiased mass spec analysis of the secreted factor when the embryo is in contact or not with the maternal tissue, and how that communication can vary when the two come into contact. And finally, the last, I could stay here till tonight to tell all the research that everyone is doing in the lab, so I have to cut and select just very few. So the last project I want to just mention is about also the genetics and the possibility now that we can do forward genetics to try to understand how a loss of function can impede these interactions between the embryo and the maternal tissue. And this is really has been taken on board by Magdalena, postdoc in my lab. And we have few candidates already, but also there are many unbiased candidates that we're trying to pull out of many other screening. But the idea is to use CRISPR. In this case, we want to use Cas12 and not 9 to have more precision in the editing and also less off-target effects. But the project is to both induce gene loss of functions in the embryonic compartment. In this case, we cannot do in the human embryo, so we use blastoids, which are stem cell embryo models, as the best proxy of the human embryo. So we induce this in the cells, and then we can make basically knockout mutant blastocyst-looking structures, and then test how a loss of functions in the embryonic compartment induce a potential phenotype in the implantation when you have a normal endometrium, or vice versa. We can induce the knockout in one of the endometrial cell type of the maternal tissue and keep a complete, wild type, either embryo or blastoids, and then see how the loss of functions in the maternal tissue can affect that interactions. So this is the last slide. So hopefully I hope I'm on time. So I wanted to thank first all the funder that allowed this, but especially the very first grant I got is the MCHRI grant. So that's really a big thank to MCHRI to be part really of this journey from the inception. And here I want to thank everyone in the lab. So it's a growing lab, as you can see. I keep shrinking the pictures because now adding the people, the slides is, so I really have to make space. I need to find a way now to fit everyone here. And then lastly, a big thank to the people who recruited me and believed in me and my projects and keep supporting me: Leslie, Virginia, Yasser, and Ravi from OBGYN as well, and the Institute of Stem Cell. Thank you very much.
Thanks, Matteo. Very nice talk. So you mentioned maybe the problems of the endometrial vessels building into your platform, but also my question was about the extra embryonic vessels. Do you think you'll be able to assay that aspect of sort of implantation?
From the fetal part?
From the fetal side.
So I thought about that. So because of the culture, the legal limitation that we have with the human embryo, we cannot get into vasculogenesis from the fetal side because we cannot pass the gastrulation point as a kind of. But there are also lots of discussions about changing the rule here in California, at least. Even it's happening in London, but as you know, London Parliament is a little bit. It's going to take time considering the legislation that it has there. Here, it may be possible that if they change that law, which may happen soon, we'll be able to look at later stages and look at initiation of vasculogenesis, because my dream is going to be to be able to access the stages of heart development and especially neural tube development up to day 28, which has been my proposition to the California Institute of Regenerative Medicine. And hopefully we can go there so we can study these two important stages of heart development and neural tube development and all the congenital defects associated. So I'm hoping that this is going to become true as soon as the law allows us.
Okay, great. Well, really exciting progress.
Thank you. Thank you.
Thank you. Really, really incredible work. I was surprised to see so many of the VEGF factors in that specific day 14 SOL. So Dr. Mackenzie was our visitor for this session, and Dr. Mole is our internal recruit. And our third speaker is somewhere in between. So Sheng is currently a professor of chemical and nano engineering at the University of California, San Diego. And he is here to talk about wearable ultrasound technology for fetal and maternal health monitoring.
Thank you very much. So good afternoon, everybody. I know it has been a long day. You have been absorbing lots of information. So thank you very much for staying until the last of this session. What I'm going to show you in the next 20 or 25 minutes is something that I guarantee you do not see at any other place. This is also the first time I'm presenting publicly. So let's see. So we work on those wearable ultrasound technology for maternal and fetal monitoring. And I don't think I have to preach to the crowd that this oxygen for the fetus is so critical. And oxygen, the only source of oxygen to the fetus is through this mother to the placenta to the cord and to the baby, right? And as you can probably imagine, if there's any disruption of those blood flow to the fetus, for example, due to this nuchal cord or cord compression, then all kinds of complications will happen, right? Such as like this, hypoxia and cerebral palsy, and the most adverse example of this case is stillbirth, right? And unfortunately, stillbirth is largely unknown. People do not know what's going on, what's the cause of it. And the best we can do right now is to do postmortem analysis, to do this histology study of the placenta, so on and so forth. That means what? That means baby is really a black box, right? So how can we probe into this black box to understand the pathology, understand what's going on that lead to these bad outcomes? So this has been a very, I would say, crowded field, and there have been lots of technology to probe into this black box. For example, conventionally, people use so-called cardiotography, which basically is a device that can monitor your fetal heart rate, monitor the uterine contraction. And for example, another example is this electrocardiology, right? And that monitors the maternal EKG and also sometimes you can also monitor this fetal EKG, right? So those devices have limitations in challenges to differentiate maternal and fetal signals, and also the signals fluctuate a lot during those maternal and fetal movements. And most importantly, those devices only sense signals on those maternal skin surface. It only scratches the surface. It does not go deep. There have been some newer versions of those sensors, but those sensors are pretty much the same as those conventional devices. They are basically old wines in new bottles, and they have similar challenges as those conventional devices. So when you talk about those fetal monitoring, I would say the most commonly used modality is ultrasound. Why ultrasound? Because ultrasound can probe directly into the fetus, right? It can extract lots of information. And what you see on the left is the most commonly used ultrasound is a cot-based, and it can be very functional, very powerful, but it still has these limitations in discontinuity, which means you can only do this monitoring on a sporadic cases, like once in a month for those pregnant participants, right? And this is highly user dependent, which means it requires a sonographer after many years of training and lots of experience to do it well. And different sonographer may give you different results. And because of these requirements of this sonographer, this diagnosis relatively expensive. It's not the problem for Palo Alto, not the problem for California, but for those healthcare deserts and low middle income countries, this is a significant challenge. So our vision is to re-engineer this ultrasound card into a ultrasound patch. It's like a sticker. And this sticker will be able to adhere to the abdominal area of the mother, and then those electronic controlled ultrasound beam will enable 3D scanning and imaging. And those AI-based algorithms will analyze the results and tell you those actionable information when there is any sign of those risk or danger to the baby or the mother, right? And the benefit of this, ultrasound sticker is that it is continuous. This patch is low form factor. It can provide you 24/7 continuous monitoring. You do not lose any data. And this is user independent because in this entire process, you take this ultrasonographer out of this picture, right, with minimal input of those experts. And because of this is low cost, it lowers down the barrier for adopting this technology. So with this ultrasound sticker, we are able to continuously monitor the blood flow in the cord to the fetus and know what's going on. Pattern change of this blood flow, and that can enable those early intervention in case any signs of danger or stress to the fetus. So what you're looking at here is this ultrasound sticker that we developed about 7 years ago. Now we can do much better than this, which I have a few examples to show you later. So this is the ultrasound patch, ultrasound transducer 10x10 array. Each one of them is individually controlled, multi-layered to maximize the performance. So at the bottom here, showing how soft, how compliant those patches are. You can stick them to the body and move around without any constraint. Does not limit any of your daily activities. So what we can do with these ultrasound stickers, right? It can do those beam mode imaging. It can measure the size of the fetus. For example, this head circumferences, this abdominal size, this femoral length, so on and so forth. So you know the fetus is small or large for this specific gestational age. And our measurement result from the ultrasound patch is validated by this clinical device. They have similar results. And additionally, we can use this ultrasound sticker to monitor the flow, hemodynamics, right? And we can image these, fetus and focus the beam to any part of the body that you want, as showing examples here, such as the circovillus in the fetal brain. Those are the spectrum Doppler showing this blood flow velocities, peristaltic and diastolic. And this is the umbilical cord. You can see the spiral geometry of this umbilical arteries and vein, and those are the corresponding blood flow spectrum from the cord. And we have validated our measurements on 62 participants. And what you see here are examples of those 12 of those participants. And you can see that those blood flow waveforms, vary, considerably depending on the condition of this, mother. And we also validated the measurement accuracy of this ultrasonic patch against those clinical device using those blend Altman analysis. You can see that most of the data points are within those 95% of this limits of agreement. So baby move a lot in the uterus, right? And when they move, those cord will also shift. How do you make sure your ultrasonic patch will monitor at the same location? Will be able to track this moving target, right? So this is where AI, where this imaging segmentation comes into play. So what you see here is an example. So at the beginning, you can see this is the cord, and as time goes by, you can see this cord, position, geometry, orientation, everything's shifting. So how do you track those moving target even without any human intervention, any human input? So we do this image segmentation and eventually know this a pulsating pattern of those vein and artery, and then we can, after we know the location of these those artery and vein, then we can steer the ultrasonic beam specifically target those vein and arteries. Therefore, we can get this blood flow spectrum continuously without any human intervention. So this is like with this AI tracking. This is without AI tracking. You can see that these arteries moved in and out of all of those ultrasound window, and therefore your recording is on and off. So here's an example showing that a case study showing that we can do this live tracking in real time. So this is the video. I'm not sure if you can play. This is the video. Can you help play this video? You can see that this is where the ultrasound patch is. This is the window, and this is the loop of this this umbilical cord, head, and abdominal area. And you can imagine a little bit this baby move around, and we can track this moving target. This is another video. Can you play? No, let me see. This is a mouse. Can you see it? Yeah, so you see that. You can see that in this video that this baby is moving around, and we can use this green dot to analyze and follow this moving target. This green dot basically means where the ultrasonic beam is steering and focusing on, right? And this is the blood flow waveform that we can record in real time. So then the question we have to answer is why we need wearable patch? Why do we need to monitor the fetus continuously, right? So why not these monthly checkups to the hospital? So for some conditions, they have those sporadic transient behavior that may be missed by those sporadic, monthly check, right? And the interval between them is so big, you will miss some of those, significant, signs. Here's an example. For a healthy mother, you can see that those, fetal heart rate and also the resistive index are very normal, right? You can see those waveforms, are very, normal to those, you cannot identify any abnormalities. But for a mother with, identified preeclampsia, you can see that these, mother have those sporadic high resistive index, and that is reflected by this absence. You can see that this is a systolic velocity, this is end-diastolic velocity. For some cases, this end-diastolic velocity will disappear. That means what? That means the flow to the fetus is not sufficient. And that can be a significant concern for the fetus. And this is a real example. This is one example of our study of this 62 cases. Once the student identified this absence of end-diastolic velocity, he notified the nurse, and the nurse discussed with the doctor, and eventually they put this mother in the ICU. After 4 days, they decided to do C-section. to deliver this baby. So as you can tell here, using this conventional approach, using this fetal heart rate is not any difference from those healthy patient, right? Healthy participant. So this is the summary of the results. You can see that this is for the fetal heart rate, and for the preeclampsia patient and the healthy participant, they have a pretty similar fetal heart rate. But for this resistive index that can be measured by our wearable ultrasound patch, you can see the preeclampsia patient is very, very different, much higher than those healthy participant, right? So this is just one example. So we summarize our results. So 62 patients for those study, but we continuously monitored only 52 participants. You can see that for fetal heart rate, you cannot really tell any clear trend for those as a function of gestational age. But with this pulsatility index, you can see that it has a clear trend, right? It's gradually reducing, as gestational age increase. Similarly, for those predefined those perinatal risks, such as healthy, small for gestational age, large for gestational age diabetes, preeclampsia, hypertension, for fetal heart rate, you cannot really tell them apart. But with this pulsatility index, you can clearly identify those potential risk concerns. People may ask, well, if you put this patch on the abdominal area and continuously, shine ultrasound to the baby, are there any safety concerns? The answer is no. So we have done those very, very careful and a professional this characterization of the ultrasound intensity by working with a ultrasound expert at FDA. And as you can see that this is our measurement results. The safety indexes, including this mechanic index, these thermal indexes of all kinds, are well below these regulatory thresholds. So this is for the fetus, right? How about for the maternal? How about the signals we can extract from the mother? We can measure a lot of things. For example, blood pressure, right? Central blood pressure waveforms. Here's an example. We can monitor the blood pressure from the carotid, and this is a measurement mechanism. Basically, we are measuring the vessel diameter, and from the vessel diameter, we can extract those blood pressure after calibration, right? So this is a beat to beat blood pressure waveform. Every beat, every heartbeat, we can measure 200 data points. So it's lots of high temporal resolution. We can also monitor the brain flow to the this circle villus, right? So this is for the adults. You see those 3D this blood vessel mapping through the temporal window. You can identify all those major branches of those cerebral vessel, and we can pretty much record all those blood flow waveforms from those branches. They have different morphologies and directions, so on and so forth. And we can image in the heart continuously and non-invasively without any hand holding. And these are the 4 typical views of diagnosing your heart, right? This is the schematics, and this is the imaging result from the wearable patch, and this is the imaging result from a clinical device. You can see similar quality. We can also have those different views if you imaging along this personal short axis views and the 7 segment model, and there have been evidence to show that this mechanical motion, mechanical beating, this regional wall motion abnormality is about half an hour earlier than the EKG to predict heart attack. And lots of data, lots of images, right? How do we deal with it? You guys are very busy, so we worked with data scientists to develop this AI model. And in this case, we can use FCN32 model to extract those left ventricle volume, and from left ventricle volume we can derive stroke volume, cardiac output, and ejection fraction. And I'm sure I don't have to preach to the choir how important those cardiac indexes are. So this is the left ventricle volume waveform, and from this zoomed in 3 critical moments, you can see that those waveforms are very different after exercise. So this is high heart rate, slow heart rate. You can see this waveform morphology is also different. You can see this dichroic notch, and from this left ventricle volume waveform, you can derive ejection fraction, stroke volume, cardiac output, and heart rate. And ultrasound is a mechanical way. We can derive those mechanical properties of human tissue. And biomechanics is related to lots of disease, cardiovascular disease, cancer, aging, fibrosis, so on and so forth. So the student did a experiment by frying a steak from the bottom. You can see as a function of time. So the bottom, the stiff part is actually growing thicker, and the interface is moving up. So ultrasound can also capture those chemical information. So in this case, we can image this hemoglobin distribution in the human body. This can be useful for detecting this breast cancer and the hemorrhage. So in this case, we use a laser. Laser shine into the human body. Those molecules absorb the laser. Then they swell, and when they swell, they emit ultrasound. And you can see that we can use conventional ultrasound. You cannot really tell them apart from this human breast cyst. But with photoacoustic, you can see that blood can be clearly differentiated. Using different wavelengths of those lasers, you can sense different molecules, different chemicals. So photoacoustic signal amplitude is linear proportional to temperature. Therefore, we can do 3D core body temperature mapping, right? So this is a Fenton experiment, a warm, cold region, somewhere in the middle of the temperature gradient. And those are the two thermocouples. You can see the thermocouple reading and the photoacoustic reading matching each other very well. So what are the benefits of being wearable, right? For ultrasound, for conventional ultrasound is powerful, but it's manual, requires experience of the user. This is operator dependent. It's expensive, as I just said, and this is a one-time testing. Happens in only hospital, gives you only snapshots. There are lots of data, information missed, and the patient has to remain stationary. This requires gel. And for this wearable patch, you can see that it's hands-free. Just put it on any part of the body, and the electronic scanning data analysis by AI can empower general user to use it. It's operator independent. It's low cost. Everybody can use it, right? So this is continuous anywhere, anytime. It gives you all those comprehensive data results, and most importantly, you can couple stress and motion to this entire imaging process. This is gel-free, right? So because it's a patch, it's like conformal to the human body. So this is my group, and this is students and postdocs. I'm learning from them every day. My critical collaborators so far, funding agency. The size of the logo is roughly proportional to the amount of funding accumulated over the years. So so this is exciting. And what I did not mention so far is that in 9 days I'll be joining Stanford Department of Anesthesiology. Look forward to working with you all. Thank you.
Thank you very much. Really amazing technology, and welcome to Stanford. I'm curious, for the pregnancy application, do you envision teaching the mother to read the output from the Doppler or ultrasound? Will the mother be engaged in the monitoring?
Very good question. So I think that's a very complicated question. I think to simply put it, we can simplify the readouts, like give those mothers 3 levels, like red, yellow, green, so that when there is a significant concern, they're encouraged to go to the hospital to seek for a more professional advice.
Is this something you tested in patients to see how they respond, how their well-being during pregnancy is affected by potential false positive signals?
Very good question. We have not done those customer survey yet, but we in our IRB studies, we did provide them some data sheets, so to provide feedback on how friendly those technology is and anything to improve, so on and so forth. Yeah, I think we should definitely engage more those end users. Yeah.
Thanks.
Thanks so much. Yeah. Thanks, Chris. All right. So we've come to the end of the day. Thanks for hanging in there. I wanted to first thank the education committee who basically helped put this on. They're in the room here. I just want to give them a big hand and applause for the education committee because it's been a lot of time together. There's also 2 individuals who are still here in the back that I wanted to have stand up. Mary Chen and Grant Wells, can you please stand up? These are the people that make it happen. Please thank them on the way out because none of this could happen without Mary Chen and Grant Wells. And also for you, thanks very much. I hope today, just to summarize, We're learning about what's missing in maternal and child health. I think we learned a lot about that today. From the 3 sessions we had plus the speakers, there's a lot, genetics by environment. There's a lot of genetics we can understand. There's a lot of environment we can understand. And our speakers today really basically set the bar for what things can do. And also then we learned in this last session, we actually have therapies now, personalized surgical therapies, and also modeling that we can do to basically make it happen. And so hopefully in the future, we'll be able to do that. So thanks very much. There's some drinks in the lobby, so please help yourself, and thanks for coming today.